Thanks for coming to my talk. This talk is part of the Robinson Medal lecture series sponsored by the Geological Association of Canada's Precambrian division for 2020. And of course, that was after the pandemic started, which explains the delay in my delivering the lecture tour until now. This talk explores a scandalous little paper that I published last summer, in which I suggested that I may have found evidence of the oldest known animal body fossils. Why was it so scandalous? Well, the material is around 890 million years old, and I'm suggesting that it may be fossilized sponges. That's more than 300 million years older than the next youngest undisputed body fossil. So you can see where this might be perceived as a brazen claim. So at this point, I want to emphasize that in everything that I've said or written on the subject to date, I've used equivocal language and subjunctive verb moods. And that's because I want to be clear that although I think I have compelling evidence for what I'm arguing, I don't need to be right. This is the beginning of a discussion over a fresh approach to the question of animal life in the Precambrian. And my hope was, and is, that the ideas that I'm suggesting can form the nucleus for a collegial, dispassionate, evidence-based discussion in the scientific community involving many types of relevant specialists. 160 years ago, On the Origin of Species, Darwin was blunt about what he thought was the most concerning weakness in the evidence supporting his ideas about evolution: the seemingly sudden appearance of taxonomically diverse mineralized fossils in the Cambrian and the absence of any such material prior to what we now know as the Cambrian explosion. Earth is 4.5 billion years old, and the most important time boundary in Earth's history is what's informally known as the Precambrian-Cambrian boundary. Right here, I'm pointing in the upper left. And this boundary separates the Phanerozoic, so the time of visible life populated by a profusion of readily intelligible fossils, from the Precambrian, a time that contains no normal fossils. And this rapid transition, known as the Cambrian explosion, was not only an expansion in animal diversity, it was also an explosion in animal preservation, and resulted in the appearance of animal hard parts, what we think of as normal fossils, so shells and exoskeletons and the like. And so it's apt that the third eon in Earth's history, the Phanerozoic, the last 10% of Earth's history is called the Phanerozoic, because phanero comes from a Greek word that means to become visible. The science of fossil preservation is called taphonomy, from the Greek word for the grave, and it's one of the topics I'll be talking about today. And a lot of how we think about organism preservation in the rock record is quite biased by the ways in which regular issue body fossils like these ones are preserved. However, there's a number of ways in which the soft parts of animals can be preserved too, even though statistically soft-bodied fossils are quite rare. So here is a modern genetically based universal tree of life with the hypothetical first cell somewhere in the middle. And on this diagram, the spoke length and the grouping of the spokes indicates degrees of genetic relationship and difference. It indicates natural evolutionary groupings among major groups based on genetic information. So in this group, in this view, there are three so-called domains of life. Domains bacteria and archaea are what, those organisms that we would colloquially call bacteria. Single-celled organisms whose DNA is not packaged into nuclei. They have a different level or a different kind of cellular organization from domain eukarya down here in the bottom, which are those organisms that have membrane-bound organelles inside their cells. So things like nuclei, mitochondria, and chloroplasts. And it turns out that eukaryotes are actually not exactly their own domain anymore. They seem to form a subgroup among the archaea. Domain eukarya contains or includes everything that we would consider normal organisms, such as, plants and animals, fungi, algae, protists. But as you can see, all of that is just a tiny part of the diversity of life. However, we are eukaryotes, and we have an outside-sized interest in ourselves and in where we came from. So research on the early evolution of eukaryotes is a major topic these days. Among the eukaryotes is a very particular group, this little green text down here in the bottom, called the opisthocons. And the opisthocons is an evolutionary group that includes both fungi and animals together. So In other words, of all the other eukaryotes, fungi are our closest relatives. When we think of major biological events in deep time, we can't consider life in isolation from global events. Life first appeared sometime in the Archean, and one of the important things it eventually did was contribute oxygen to the otherwise oxygen-free atmosphere and ocean through photosynthesis. Oxygen is a critical substance for animals. We require a certain minimum concentration of oxygen in the atmosphere and ocean water. And so although there's an awful lot of attention given to the great oxygenation event in the Paleoproterozoic, there is a second, less well-known oxygenation event that is probably much more important, at least as far as animal evolution is concerned. So the Neoproterozoic oxygenation event, which I've exploded over here on the right, it's kind of mysterious. Some people think it took place in the Ediacaran. Others find evidence as far back as the Tonian for the Neoproterozoic oxygenation event. The main point is that there's a general idea that animal evolution couldn't get started until a certain minimum level of oxygen, a critical level, had been reached after the Neoproterozoic oxygenation event. Okay, some other events that may or may not have been important to the evolution of animals prior to the Cambrian are the cryogenian glacial episodes, the so-called snowball Earth episodes, which, depending on their severity, may have precluded animal evolution or at least influenced it in some way. So there's been a lot of attention through the latest parts of the Neoproterozoic for a variety of reasons, among which is very conspicuously Darwin's Dilemma. Animals, or metazoans, are multicellular eukaryotes that ingest, in other words, eat organic matter to get their energy and nutrients. And the animal tree of life looks something like this, depending on whose work you refer to. This phylogenetic tree is based on molecular data, so work that analyzes genetic information to identify evolutionary relationships. And the main groups of animals are the porifera, or sponges, the cnidaria, that's corals and jellyfish and things like that, and bilaterians, okay, animals, everything that has bilateral symmetry like we do. And it's now generally agreed that sponges are the most basal form of animal, that they are a sister group to all of the other animals, and that they probably emerged first evolutionarily. There are a number of reasons why it is reasonable to think that animals were present in the Neoproterozoic, foremost of which was, or is, Darwin's logic. It just doesn't make sense that so many complicated animal types should spring into existence in a geological instant. Surely there was a prehistory, some sort of gradual hidden evolution that preceded the seemingly sudden appearance of mineralized animal fossils in the Cambrian explosion. Molecular clock research takes the relationships identified by molecular phylogeny and uses the known rate of mutation in certain types of genetic material to back calculate the time when major lineages of animals and other organisms emerged. I'm certainly no expert in this. Molecular clock work requires that major divergence points of major lineages be calibrated. And of course, the best way to calibrate would be using the fossil record. But here, of course, in the Proterozoic, we encounter the problem that the absence of evidence is not evidence of absence. And so molecular clock work for estimates of major evolutionary events in the Precambrian, which is where most of them probably took place, understandably have a lot of uncertainty. Nonetheless, a growing volume of molecular clock work suggests that sponges probably emerged sometime in the early Neoproterozoic, in the Tonian. And people have been looking for physical evidence of sponges in cryogenian and Ediacaran rocks, but so far conclusive evidence is lacking. The Neoproterozoic does not exactly lack large and or complex fossils. There are a number of complex things that are already well known, including the Ediacara biota, the small shelly fossils, and even putative animal embryos. But nonetheless, all of these things are ultimately not undisputedly animal related. And anyway, they're all limited to the Ediacara. there is a growing body of evidence showing that eukaryote communities were already quite complex by about 1 billion years ago. And I'm drawing your attention to this recently published almost 1 billion-year-old fungus fossil from northern Canada, whose evidence or whose existence shows that the Opisthoconte lineage was already present almost 1 billion years ago. Okay, so fungi are metazoan adjacent. They belong to the same group. And last, but definitely not least, biomarkers, which are preserved relict lipids, which can be distinctive to a particular taxonomic group. Biomarkers affiliated with demosponges were reported from cryogenian strata about a decade ago. And although this work was quite controversial at the time, the evidence and the rationale seem to have withstood the scrutiny since then. So given all of these suggestive lines of indirect evidence, let's explore some possible reasons why Darwin's dilemma has not been resolved. First of all, we can completely dismiss the idea that complex and or large organisms were totally absent or not possible, okay? Second, what about the idea that animals existed in the Proterozoic but simply weren't preserved? This would imply that preservation was not possible, either owing to something to do with the fossils, or sorry, to do with the animals themselves, or something to do with the conditions under which they died and were buried. And if this is the case, we're not going to be able to resolve Darwin's dilemma. But before we concede on this point, there's an absolutely gargantuan amount of work to do, because actually, I would argue that we have barely scratched the surface of possibilities. And my own perspective is that we may have been too biased in what we are looking for and looking at. In other words, what we consider to be eligible in our search. So what do I mean by biased and limited? Well, in terms of rock type, paleoenvironment, age of rocks, type of organism, and taphonomic process, there may be a vast amount of work yet to do, given what we may have neglected or overlooked. So maybe we need to rethink our approach to this problem. So we have to understand that the evolution of early animal-like organisms in the time prior to the advent of normal fossils would not necessarily have had any of the characteristics of familiar animal phyla. Okay, and so we need to address how early animal-grade tissue may have expressed itself in sedimentary rock prior to the advent of morphologically intelligible phenomena such as trace fossils and body fossils. And this means thinking outside the box when it comes to preservation. Question: are there taphonomic pathways we haven't considered? In the search for Proterozoic fossils in general, there are two rock types that are most productive: mud rock and chert. Other rock types are generally not the focus, and that includes carbonates. And although carbonates are generally mineralogically simple, they are among the most texturally complex of rock types. Unlike all other basic rock types, the nature of carbonate rocks is very strongly time-specific, and that's owing to their intimate connection to Earth's dramatic biological and geochemical evolution through time. They precipitate from seawater under the influence of organisms, and they encode the fluid's geochemistry in their crystals. On top of that, there's the fact that carbonate rocks are extremely prone to post-depositional changes, in other words, diagenesis. So all to say that carbonates are intimidating, and Precambrian carbonates are especially problematic because it's hard to apply uniformitarian principles to a world that's so different from what we're familiar with. Another interesting reality is that a very logical approach to the question of Precambrian animals is, well, we should push back incrementally in time from what we already know. And so most of the work to date has been on the Ediacaran. And perhaps it's time that we reconsidered our biases and assumptions in this regard. Also, given what molecular clock work may suggest about the potential for animal life prior to the Ediacaran. And last, I want to emphasize that we're animals that are good at pattern recognition. So we're instinctively looking for something that we already know, that we recognize, things that are records, in fact, of fairly complex animals. And perhaps this isn't the way to approach the earliest animals, which could have been quite dissimilar to anything we know. We should be looking for the simplest version of the most basal animal. But what would an animal simpler than our current idea of the most basic known type of animal look like, and how would it be preserved? Do we even know how to look, how to think about that? So maybe it's time we take a closer look at sponges. So the material I'm eventually going to expose you to is from Tonian rocks, okay, much older than commonly considered possible for animals. Carbonate rocks, generally not considered propitious for Precambrian fossils. And it represents a type of taphonomy that until now has simply not been on the radar screen at all. So to start off, we have to have a good hard look at what it means to be a sponge. The classical depiction of sponge anatomy is of a cylinder or a cone-shaped body. Here's a cross-section through the wall of a sponge. The animal filter feeds by sucking water through tiny pores in its outer surface and exhausting the water through a central cavity. Okay? Sponges have only a few cell types. On the outer surface are protective cells through which there are pores, pore cells, that lead to a system of canals and chambers inside the sponge body. Inside the canals and chambers are choanocytes or collar cells. They have, each of them has a flagella that they use to create a current that sucks water through. And water that is, or organic matter that is suspended in the water is then trapped by microvilli. And then that's the collar part of the collar cell, and then shared with other cells. Amoebocytes, these pink things, or archaeocytes, have a variety of roles, including food processing. And sclerocytes, shown here in blue, secrete spicules, shown in gray. And spicules are tiny mineralized spikes that, if present, form a flexible mineralized scaffold within which the cells and the connective tissue of the sponge are suspended. Down here in the green is are spongocytes, okay, which secrete spongin, and this is really important. Spongin is a very tough kind of collagenous protein that forms the skeleton of some sponges, okay? And all of this is embedded in a non-cellular gel-like substance, a type of connective tissue that is formed in part by a different, less hardy type of collagen. Collagen, just for the record, is the main component of the connective tissue in all animals, so it's a really important idea. Not all sponges have the standard erect cylinder configuration, the typical barrel sponge. Some sponges exist as thin veneers of tissue on hard surfaces. Some are endolithic, the so-called boring sponges, not very boring at all. Most of their body occupies canals and galleries that they excavate into hard substrates like coral or concrete piers and pilings. Rather importantly, this talk, there are cryptic reef sponges. That cryptic means they encrust the walls, the ceilings, or the floors of dimly lit cavities just below the exposed reef surface. Right? And without getting into the thorny topics of sponge phylogeny and sponge taxonomy, sponges are basically divided into 4 classes. The Hexactinellida, Calcarea, and Homoscleromorpha have spicules, have a skeleton made of spicules, either silica or carbonate. Okay, the demo sponges, which is the most diverse group today, have skeletons that are variously made of spicules and or spongin. And importantly, there are demo sponges that have a skeleton that's made only of spongin. What do sponges look like in thin section? Okay, so most problems. Here is at the bottom a spiculate sponge preserved as an intact body fossil. So the spicules are all these white things. And they're not all that rigidly fixed to one another. Sponges generally decay and fall apart after their death, so their bodies seldom get preserved. But the individual spicules, shown here in scanning electron images, they can remain and be preserved in sediment. Commonly, preservation, as I said, is not good. So you might get, at the best, a fragment of sponge and the rest of it collapsed. So on the right is a bit of sponge. You can see the reticulate spicule scaffold, and in this sort of brown, the chambers or the canals in between. And one thing that has long been just a fact of life for people who deal with fossil sponges is that they commonly have accumulations of peloids, okay, between the skeletal elements, between the spicules. And the word peloid refers to a round or oval silt-sized particle of carbonate mud. So that seems to be the story for spiculate sponge taphonomy. Spicules, maybe some peloids. However, it turns out that there is another type of physical evidence for the former presence of sponges, a type of sponge taphonomy that nobody has addressed yet in the Precambrian. There are clever spongiologists out there who work on how sponges can be preserved in the modern world, not in mud rocks or chert, but in carbonates, and no spicules are required for this. Turns out that sponge soft tissue can be converted into some fairly enigmatic carbonate mud textures when the collagen that holds the cells of the sponge body together gradually decays. Chemical changes associated with decay actually trigger the precipitation of the mineral aragonite. So in these secondary electron images in the top, the thin, stretchy looking strands are the collagen, the extracellular collagen in between the cells. And the slightly larger straight rods are aragonite forming on the decaying collagen. And backing off a little, looking in the lower right, you can see that some of the rods form clumps, which potentially could form into peloids. The upshot of this is that the soft tissues of sponge bodies can become partly calcified, permineralized, if you will, leaving finely crystalline carbonate where at least some of the soft tissue used to be. And in the rock record, this would look like nebulous carbonate mud occupying the space where the soft tissue used to be. And it may even retain the microclumpiness that you can see in these lower right images. Now, if this process of permineralization is effective, it can even produce semi-intact sponge bodies that are known as sponge mummies. Now, what would happen if you had this kind of permineralization happening in a sponge that had a spongin skeleton? Okay, spongin, remember, is much tougher than the collagen in the jelly-like extracellular matrix, so its decay rate will be much slower. However, it stands no chance of being preserved as hard parts because it's still organic matter. What about other possibilities, though? Molds, for example. What kind of a microstructure would you get if you permineralized the collagenous extracellular material of a sponge that had a spongin skeleton? As I mentioned, the most abundant sponge class today are the demo sponges. Okay, many demo sponges lack spicules and have a flexible skeleton made of spongin. These are informally called keratose sponges. And I give you the humble bath sponge, this specimen from under my bathroom sink. A bath sponge is the sponge's spongin skeleton after all the soft tissue has been removed. Okay, here's a tiny chunk of my bathroom sponge in which you can see, and this is really important, that the spongin skeleton consists of a three-dimensional polygonal spongin meshwork. This is just in incident light. And importantly, this three-dimensional meshwork consists of an anastomosing meshwork. In other words, it branches and then rejoins in 3 dimensions. This is very complex and it's very distinctive. Okay, so I epoxy impregnated my souvenir bath sponge and thin sectioned it. Okay, so you get the general idea. So down here, 30 micron thick slice shown in transmitted light. You can see the fibers that branch and then rejoin to form a polygonal 3D meshwork, you can kind of see a slice of this where the 30 micron thick thickness of a thin section is adequate to capture some of this anastomosis. So now imagine what you would get if you permineralize the soft tissue, right, encasing the sponge in meshwork. So the soft tissue is what's here represented by the gray, which is epoxy. So imagine what you get if the gray was actually permineralized carbonate mud, and it molded these spongin fibers, and then the spongin eventually decayed. You would wind up with a system of tubular molds surrounded and defined by finely crystalline carbonate that replicated the configuration of the spongin meshwork as molds. And importantly, given the size of the fibers and the spacing of the branching, a 30 micron slice through this thing, through this microstructure, would be quite capable of depicting the three-dimensional anastomosis of the meshwork. Something resembling this was already identified in the rock record almost 120 years ago. This is a very unusual microstructure, first described by Geology in 1906 in Carboniferous stromatolites that from Belgium, and he called them spongiostromids. This microstructure is quite complex, consists of little branching tubes 30 microns in diameter, make a complex 3D meshwork of calcite-filled tubes enclosed by microcrystalline calcite. And in the stromatolite literature, this is misleadingly known as vermiform microstructure. Of course, verm means worm, and it has nothing to do with worms. So Geology recognized that this was too complex to be made by bacteria, and he suggested, based on no evidence at all, that it may have been made by protists. However, what's important is that he did recognize that this microstructure does not belong in the same category as ordinary microbial stromatolite microstructures. And it turns out now that this microstructure is all over the place in Paleozoic carbonate rocks, especially reef rocks. And there are some intrepid paleobiology folks who are bravely suggesting that vermiform microstructure is in fact a type of sponge body fossil. So here in the upper left is a putative sponge body fossil encrusting the roof of a reef cavity. Lower left, here's a putative sponge body fossil living inside a cavity made by a brachiopod. And if you zoom in on the lower right, it has vermiform microstructure exactly the same as in my thin section bass sponge. But wait, there's more. Here's a Cambrian rock from Western Newfoundland, upper left. Looks like a stromatolite, right? Actually, that's only partly right. In thin section, there is a very complex, distinctive microstructure that's too complex to be made by cyanobacteria. And it has, lower right, exactly the kind of three-dimensionally branching and rejoining configuration that spongin has in keratose demosponges. Okay, so this color-coded tracing of the slab shows the spongy bits in red and the microbial bits in blue. And these authors are proposing that this is a consortium between sponges and microbes. So feast your eyes on these things, my friends. The authors of this paper interpret this as the body fossil of, or body fossils of keratose sponge that built something that is superficially indistinguishable from stromatolites. Okay, so there's now a growing but controversial body of work that considers vermiform microstructure in Phanerozoic stromatolites and other microfacies to be sponge derived. And of course, I'm here to tell you that I found exactly the same microstructure in early Tonian reefs from northwestern Canada. And I found this microstructure in only a few of the samples that I collected for another purpose altogether, understanding Precambrian stromatolite reefs. Full disclosure, these reefs were from my PhD project some time ago. Here is the microstructure intermingled with micritic microbialite microstructure. Okay, it's complicated, it's familiar, we've seen this already. Okay, so let me tell you a bit about how and where I found this material. They're part of the stone knife formation of the Little Dow group, an early Tonian bedded carbonate and shale succession in Northwest Territories. These rocks were deposited mainly below storm wave base and mainly below the photic zone. But the stone knife formation also contains enormous reefs shown in blue. These are called the Little Dow reefs, and they had substantial paleotopographic relief above the seafloor, many tens of meters or more, and they kept their growth surfaces in the photic zone. They were built by photosynthesizing cyanobacteria, and their depositional age is about 890 million years ago. Okay, so just to make sure we're all on the same page, really having mouse troubles. A reef is a biological construction that's made at least in part by organisms. It has to be topographically elevated above the surrounding level bottom seafloor, and it's mineralized from the get-go, so it's rock right from the start. Reefs are the most ecologically diverse environments on Earth, and they've presumably always been that way. That's because they offer such a huge variety of sub-environments for different reef builders and reef dwellers to occupy. Reefs have been made by a huge variety of different organisms through time, not always corals. And in the Precambrian, the first 90% of Earth's history, reefs were made by cyanobacteria, and we call these reefs stromatolites. Okay? Stromatolites are the most conspicuous evidence of Precambrian life in the rock record. And they were built mainly by cyanobacteria, which Andy Knoll called the heroes of Earth history because they transformed Earth from an anoxic to a well-oxygenated state, thereby promoting all kinds of life. But stromatolites themselves are quite difficult to understand. Very, very beautiful, but they're hard to understand. They're important. So let's have a little look at stromatolites. One of the basic characteristics of stromatolites is their macrostructure. In other words, the shapes of the bioherms, the shapes of the columns or domes, their branching, their convexity, their walls, the relationships they have with sediment surrounding them. And to me, it's safe to say every stromatolite is different. They're glorious. There's a huge range of configurations. If you ever have to work on stromatolites, I'd like to make a plug for this wonderful book, recently published, crammed with accessible information gleaned from many, many decades of many, many people's work. Lots of beautiful photos, and it's also a free download. Another basic characteristic of stromatolites, and now we're getting to the really impenetrable stuff, is their microstructure. What the laminae look like in thin section, the tiny little layers. And there's a huge array of different ways that stromatolite lamination appears in thin section, but they almost never preserve the organisms that made them. So in spite of all that early work done by people all over the world for many decades, the reality is that for most microstructure types, like the ones shown here, there's just not enough information to say anything about the communities that made them. So, you know, for the lay person, most stromatolites fall into one of two categories. Microstructures that have irregular, diffuse micritic laminae that are readily identifiable as biologically influenced. And microstructures that are very evenly laminated, each layer of a uniform thickness, commonly with conspicuous pale layers of cement that are possibly not all that biologically influenced. And in neither case is there any direct physical evidence of whatever microbial mats or biofilms were involved in the construction of these stromatolites. So in the vast majority of stromatolites, the perpetrators are just plain not preserved as identifiable microfossils. Fortunately for me, however, those reefs that I worked on for my PhD so long ago do have an interesting, unusual microstructure and an array of really beautiful stromatolite morphologies. But I had to work really hard to get that information and then to decipher it. So the reefs form gigantic pinnacled exposures and unscalable cliffs. The exposures give you no hint whatsoever of what's inside them. You can't see the microbialites on naturally weathered surfaces at all. They're just uniformly gray, prickly, incredibly steep surfaces. And so I was really flummoxed about how to put together a story of how to understand these reefs, what they were made of and how they grew. And the only solution at the time seemed to be to take a lot of samples and to bring them home and cut them open. Hope to piece together at least a very crude picture of the reef's components and their growth. And so that's what the vigorous, foolish young version of young Turner did. So I made hundreds and hundreds and hundreds and hundreds of extra large thin sections by hand. So these thin sections are 3 times the size of a normal thin section. That's because a small thin section is just not at a scale to capture the things that turned out to be in these reefs. And it was pretty desperate and kind of soul destroying, but in the end, it did kind of sort of work out. The reefs had a and this slightly embarrassing long ago diagram, I tried to depict what it might have been like to scuba dive over the reef's growth surface at different times in their growth. And on the left is what they look like in slabbed hand sample or in thin section. So the reefs had a complex growth history with a large variety of different stromatolite morphologies in the different growth phases. But one of the important outcomes of this is what the microstructure of the stromatolites is. It's not any of those garden varieties that I showed you. And instead, in this microstructure, the microbes are actually preserved. These little strands, these little dark threads, are preserved microfossils of cyanobacteria, the photosynthesizing prokaryotes that are generally assumed to be responsible for stromatolites. And when they're preserved by being calcified, they're called calcium microbes. So living cyanobacteria have slimy, thick sheaths of mucus on their outsides. It's continuously produced through their lives, and it is readily calcified in ways that I'm not going to go into here. So in this microstructure, the dark threads are the cyanobacterial filaments, and the paler stuff in between them is marine cement, okay? Probably calcified microbial sheath polysaccharide. So all of the voids in between the little filaments are filled with calcite cement that formed right away, holding this otherwise extremely porous and rather fragile microstructure together. And the reality is that most of the different stromatolite shapes in the previous slide have this microstructure, regardless of the shape of the stromatolite. Although other normal non-calcium microbial microbialites are present, they're less abundant. Okay, so the preserved part of the community that built the reefs seems to have been a monospecific community of cyanobacteria. They seem to have been aggressively growing, photosynthetic, stromatolite forming microbial mats with a vast amount of extracellular mucus. These reefs were, which then became calcified. So these reefs were as much or more marine cement as they were microbes. So here's a view of a typical view of what a lot of the reef framework looks like in slabbed hand sample. It consists in large part of irregular columnar stromatolites, these in dark gray, with intervening intercolumn sediment in medium gray, and then an abundance of marine and some later cements. This is the pale stuff, which is distributed at the tops of the spaces that separate the stromatolites from one another. Okay, these are framework voids built by the irregular shapes of the stromatolites as they grew. And here's a typical view in thin section. The main reef builders were calcium microbial stromatolites. Their irregular growth left behind framework voids, very complex voids. So these are cavities just below the reef's growth surface. They're connected to the surface and to the water column. And because of this, they can accumulate tiny drifts of sediment inside, forming geopetal structures on the bottom of each little cavity. After which, the rest of each cavity is then quickly lined with marine cement. Okay? For those who don't know, marine cement in reefs of all ages is unmistakable. It has very characteristic features that are not present in any other type of calcite cement. Marine calcite cement consists of fibrous crystals with their long axes that are perpendicular to the void wall. The fibers form an even isopachous crust over the void surfaces. And the cements are cloudy with inclusions of all types. And that's what gives them their characteristic appearance. So marine cement can start to grow as soon as the void has been formed, and they can precipitate quite quickly depending on the conditions. So as I mentioned, these reefs are especially cement rich. The Little Dell reefs were effectively cement factories. All right, let's go back to the vermiform microstructure. So I found this complex microstructure existing among microbialites. So first, let me walk you through how I know that this thing is a three-dimensional meshwork without doing three-dimensional reconstruction, which has turned out to be incredibly difficult to the point of impossible so far. So the tubules are about 30 microns in diameter. They have no constructional walls. They're defined by the size and the clarity of the crystals in them versus the darker, smaller, inclusion-rich crystals in the ground mass between them. And You can see little lines all over the place. Those are where the crystals abut one another. And because the tubes are 30 microns in diameter and a thin section is 30 microns thick, you can actually see some of the three-dimensional configuration of the tubes as they pass obliquely downward through the thickness of the thin section. So here's a clear spot where the tube is roughly perpendicular to the thin section. But here's a part going down underneath the surface of the thin section where it passes obliquely under a veneer of the darker, smaller crystals of the ground mass. You can also see within the thickness of the thin section where the tubules join and then rebranch within that limited thickness. So you can actually see the three-dimensional anastomosing meshwork of the tubules. The thing that I claim to be so distinctive to keratose and sponge skeletons. Okay, skeptics in the audience are now saying, well, surely it could be something else. What else could it be? Okay, so the most essential characteristics of vermiform microstructure are its anastomosis and the tubule cross-sectional shape. These things are 100% compatible with sponge and skeletons of keratose sponges, and they are not compatible with true algae, which branch but do not anastomose, filamentous cyanobacteria, which some can branch, but they generally, they don't anastomose, dendritic crystals, which do not anastomose and have decidedly crystallographic features, including strict branching angles, and compacted peloids, which would produce irregular gaps rather than cylindrical tubes. The idea of whether it might be fungus, I spent a lot of time thinking about, but in the end, the little dowel things don't have spores, and they lack septa in the tubules, the divisions in a fungal hypha that separate each cell from the next one. So although in a mycelium, a fungal mycelium, the older crowded parts do contain anastomosis, it really looks quite different from this. And the younger, the main parts of a mycelium, they only branch, and typically in a sort of a tuning fork-like branching. So all of this is geometrically difficult to reconcile with what I've outlined here. The most direct comparison is with the keratose sponge skeleton. Okay, let me show you some of the different places where the vermiform microstructured organism lived and where it didn't. Okay, so here's first example. It's in a shelter pore. Okay, so a shelter pore is a pore in sediment that's formed by sediment particles that lie on one another and leave open a space underneath. So this shelter pore is under two microbialite clasts. Here's some sediment underneath with some smaller chunks of microbialite in it. So in this case, the vermiform microstructured material seems to be growing down from the roof of the cavity. Okay, very nice. It note the timing of it, which is after the marine cement, the cloudy fibrous isopachous rind here lining the void. This doesn't bother me one bit, given the enthusiasm that these reefs had for producing marine cement early and very copiously. So that's example number one. Example number two is in a framework void among calcimicrobial stromatolites. So here's one of these columns. Here's another one down here. This is a framework void that is made by the construction of the reef. Okay, and so here the vermiform microstructured material, not particularly well preserved in this example, seems to be accreting up from the cavity floor. And note the timing in this case is before the marine cement, which is here filling up the tops of the last remaining bits of the cavity that's left afterwards. Example number three, it's interlayered with carbonate mud and reef debris on the proximal upper parts of the reef flank. So not where the calcimicrobial framework is present, but immediately beside it on the where reef debris was shed down the flanks of the reef. So here we have a bit of carbonate mud down at the bottom, some calcimicrobial clasts and some cement clasts shed from the reef. And here's our vermiform microstructure happily interacting with this material on the reef flank. And then last and most complicated, here are two microbial columns on the left. So here's one digit chopped off towards the left, another one right here. Okay, this is a non-calcium microbial microbialite, and the vermiform microstructures intermingled with the macritic non-calcium microbialite material. It adheres to the margin of one of these columns, and it's also interlayered with the sediment in the sediment pocket in between the two columns. And one thing that I find quite intriguing is that the ground mass of the vermiform microstructured areas is different. It looks different from both the microbial micrite and from the detrital sediment in the sediment pocket, which might support the idea that it formed in a different way, not as detritus or as microbial material, but as perhaps as a microspary precipitate. Okay, so that's what we have. The vermiform material is not at all abundant in the reefs, but where it is present, it has a very distinctive distribution. So if I put together my field observations, the structures in the slabbed rock samples, and the thin section evidence, I can come up with an idea of how this thing lived. It's clear that the vermiform microstructured organism occupied 3 sub-environments. Number 1, it was interlayered with carbonate mud, detrital mud, in non-microbial sediment that accumulated in little depressions on the reef tops and in the proximal upper parts of the reef flanks, but it is never present in the level bottom sediment away from the reefs. Lots of thin sections out there, no vermiform microstructure. Second, it occupies framework voids that are left by the construction of the reef by calcium microbial stromatolites. Okay, so it's living in little nooks and crannies, but is never interlayered with the calcium microbes themselves. And third, rarely, it is intermingled with several types of non-calcium microbial stromatolite laminae. So here's what I'm suggesting it means. This organism lived only in the vicinity of the reef building community, which consisted of oxygen-producing photosynthetic microbes. The reefs were probably oxygen oases. Okay? Possibly the organisms needed the oxygen provided by the cyanobacteria that were photosynthesizing copiously on the reef tops. Okay? Animals need oxygen. That's thought to be one of the main factors limiting the timing of the appearance of and diversification of animals. However, some sponges, modern sponges, are known to have much lower oxygen requirements than other metazoans. So this organism, possibly a sponge, lived in those micro niches where the calcifying photosynthesizing cyanobacteria did not live. In shaded depressions, little nooks and crannies on the reef top, in cavities, and interlayered with microbialites that were not built by the fast-growing, quickly calcifying cyanobacteria, with whom it probably couldn't compete for substrate, and also in the upper parts of the reef flanks, which still would be in the favorable zone of oxygenated water. Okay, so these are in fact familiar niches for modern and fossil sponges. So in the bigger picture, perhaps it makes sense. Okay? We have perhaps the appearance of some type of protosponge in the early Etonian, which would be compatible with the predictions of molecular phylogeny. It would confirm that the products of biological evolution were able, by and large, to survive through the cryogenian glacial episodes. Okay, so no severe pruning back by the glacial events. It would suggest that the emergence of the earliest animals may have taken place in a Tonian low oxygen world, and that these organisms were therefore not tied to globally increasing oxygen levels of the Neoproterozoic oxygenation event or events, whatever it or they actually were. There's pretty good evidence that they lived in an environment where oxygen was comparatively high and consistently available. And although I can't prove this, it's just a suggestion, possibly even where abundant food was available in the form of suspended polysaccharides shed from cyanobacterial sheaths. And this is a suggestion that has also been made for some of the Paleozoic examples of vermiform microstructure intergrown with microbialites. So perhaps the earliest animals emerged in oxygen oases in an otherwise low oxygen world. Perhaps early sponges emerged in the Tonian and then lingered unchanged and fairly unchallenged by evolutionary pressures for several 100 million years in special environments like these reefs where there was extra oxygen available. Or, maybe they were truly low oxygen animals and they lived in lots of environments, but were only preserved in these cement factory reefs. And perhaps, all of the other more complex animal types did not emerge until after the Neoproterozoic oxygenation event, when dissolved oxygen reached a level that was suitable for their more demanding metabolisms. So, as I said, I don't need to be right, although I do think I have some compelling, interesting evidence. So my hope is that my foray into this world of extremely strong opinions will trigger a collegial evidence-based evaluation of an interesting idea whose time has perhaps come. So thank you so much for coming to my talk. Hope it gave you a break from the terrible doom scrolling.